We have the ability to track threatened species through their footprints. At least that is what this article and paper that is associated with this article is saying. And this is the wonderful world of eDNA. So in today's video, we are going to cover, how these scientists are actually tracking polar bears through their footsteps, give a brief primer into eDNA, and discuss the conservation implications of this incredible methodology. Today's video is sponsored by me. Subscribe to me on Patreon if you want to see these videos a little bit earlier than everyone else and get exclusive content. Timestamps are down in the description, but first we are going to talk about this incredible news article that I found on phys.org. It's linked down in the description, and it is following in polar bears' footsteps. DNA from snow tracks could help monitor threatened animals. And what this article is talking about is the incredible methodology of eDNA. The E stands for environmental. This is getting DNA from the environment and using it to inform biology, conservation, and, anything related to wildlife. Let's first start with a brief explanation of what environmental DNA is, also called eDNA. So of course, whenever we're talking about DNA, we are talking about those wonderful double-stranded deoxyribonucleic acids. And this information forms the basis for so many biological studies. From DNA, we can of course track individuals. We can say this DNA is from this individual polar bear, from this individual frog, or from this individual bird, really any organism. But it can also reveal information about their genetic diversity. How diverse are these populations? It can reveal ancestry. How are animals related to one another? And we can even use it to estimate how large a population is. And that's with normal DNA. However, eDNA offers some key benefits over, it's the same DNA. It's really just how they get the DNA. Most traditional cases of getting DNA, of extracting DNA from an organism, are invasive or lethal methods. And what we mean by that is that we are sedating the animal and taking a blood sample. We are using preserved specimens or animals that we have euthanized and are going to become preserved specimens, and we take tissue from the liver, for example. This is, of course, a highly invasive procedure that can have lasting effects on the wild populations. Now, of course, in the grand scheme of things, by actually getting this information, we can better understand the organisms so that we may better conserve them. But what if we don't have to rely on invasive or lethal means to get this DNA? That's where eDNA comes in. eDNA, owing to its name, is getting information, genetic information, from the environment. And there are many, many different ways of doing that. In this study that we are talking about right here, following in polar bear's footsteps, what is happening is when the polar bear is stepping in the ice, when it's stepping through snow, it is leaving a footprint, okay? This is not going to be anatomically correct, and I'm assuming they have 5 toes. But whenever they leave a footprint, little bits of skin are flaked off, and inside of those little skin flakes left behind in the polar bear footprints is that DNA, because the skin does hold some DNA. So what the scientists do is they actually collect the whole footprint. It's in snow, so you can just use a shovel to pick it up, and they melt it down very gently so that just the snow evaporates, and we are left with a solution, a water solution that has the DNA inside of it. This is so freaking cool. I love DNA. And actually, so I know DNA from literally what they'll do is they'll go into ponds. Okay, so this is water, as you can see, and you can take a water sample. So you get a little beaker, you fill it up with some of that pond water, some of that pond scum, and inside of that water is DNA. And we can actually detect organisms. So say the scientists take that water back, they sample it in the lab, they test it according to some other genetic databases such as GenBank, and they say, hey, we are detecting the presence of alligator gar in this water. Hey, we are detecting the presence of amphibians in this water. We are detecting the presence of an endangered species of algae in this water. they can then say, hey, there's an endangered species here. Hey, these animals are actually present in this water, all without needing to catch these animals, needing to observe these animals. There is no need to euthanize an animal in order to get this information. Now, I love eDNA. I think eDNA is some of the coolest techniques out there, but there are some major drawbacks. Okay. One, of course, is cross-contamination. That's, of course, a huge one, and you do need to account your models for that. So your statistical analyses, your bioinformatic analyses, you need to account for that. So let's actually write down some of these cons. We'll start with the cons. So we have cross-contamination. We also have quality. So not all DNA is perfect. DNA can be degraded, and of course in an environment it can be more easily degraded, so that you need to have high quality DNA to do high quality analyses, of course. So you do have issues of data quality. You also do have many issues related to proper identification. So whenever we are working with species, we need to create primers, and we need to create protocols, and we need to create sample material, reference material for every single species. Just because we have DNA and we can analyze it, say this is from a species that has never had their DNA extracted before. How do we know that we have their DNA? Right? I mean, it's easy to hold up some information and say, this is what it is. But how do you know if you never saw the animal, if you didn't collect it directly? So these are all major cons of environmental DNA. And these are well discussed. These are well known. There's no real argument on this. But this study helped out on dismantling and further proving that some of these cons may not be as bad as we think. So, of course, the full study is linked down in the description, capturing environmental DNA in snow tracks of polar bear. They did also do Eurasian lynx and snow leopards. So this is showing that it can work on a multi-species framework, not just with polar bears. But in the discussion, there were some really interesting lines that I want to share with you. The first is that they were able to get retrieval from wild polar bear snow tracks 87% of the time. That is very high retrieval rates. For the lynx, it was a little bit lower. It was 76% from trained personnel, and then 59% from non-trained personnel, so non-experts, more of a community science framework, if I'm remembering this study properly. But, so really what they're saying is that they can get the data even from a non-traditional source like snow tracks. So this is awesome. But they really did talk about those cons that we mentioned. Post-sampling challenges of eDNA for population genetic analysis. There's many issues with amplification errors, degraded DNA, false alleles. So in essence, there are those issues with data quality that we talked about. What these authors actually did was compare it to control samples. So they collected eDNA and they collected DNA from those invasive sampling methods. And here is what they found. For all samples across all loci amplified, we retrieved identical genotypes from their snow tracks and their respective positive controls. So what this is saying in non-technical jargon is that for all of the genetic material they sampled, they compared it to ones collected, they compared it to their controls, and they were identical. So the DNA degradation issue may not be as problematic as we think, at least in this study system with this methodology. They do note that the low quantity of DNA is a concern. They didn't get a lot of DNA from the snow tracks, but the overall quality was very, very high. So there may be other errors associated with amplification of the DNA, but this is very, very, very encouraging results. And what I really, really enjoyed about this paper is that they have several sections that very clearly detail the benefits of this method for the research field, for conservation, and also detailed some next steps. So we'll talk about these very briefly. particularly for the method for the research field, that heading is a little bit vague. But in reality, what they are talking about is how these methods of extracting DNA, or really tracking wildlife in any capacity, is a cause for concern by Arctic indigenous communities. So this is a very valid concern. Indigenous communities do protect the vast majority of the world's biodiversity. And the concept of, shooting and anesthetizing a polar bear to study it, or possibly euthanizing a polar bear to study it, is a very hard sell. Additionally, many indigenous communities still practice hunting of these animals in a traditional way. And by doing studies, you may disrupt these traditional sustainable hunting methods, particularly if you are injecting the animals with anything such as anesthesia, right? If an anesthetized bear is then eaten by someone, there could be lingering effects of those chemicals inside of the body of the animal. So this is such a fantastic method because it doesn't rely on any of that. And not to mention, in the case of large carnivores like polar bears and snow leopards, this method is so much safer for the researchers themselves. They don't have to rely on interacting with these animals up close that have been known to kill humans. They can really do it from a non-invasive method. They can do it basically completely safe. And it's amazing that we have this technology. And that's what I really want to talk about, is how this can be used to save species. So what are the conservation impacts? Because I just talked about the fact that we can collect this data, but I didn't really mention how we can use this data, right? So this is likely not going to be used for taxonomic reasons. We're likely not, definitely not at this stage, going to be using this information to split species to figure out their arrangements in evolutionary space. But for the field of population genetics, this is such an incredible suite of technology, because population genetics deals with the genetics of populations. These are things like genetic connectivity between populations. These are issues like genetic diversity. How can we actually make sure that our populations of conservation concern are being, genetically conserved? So the authors here list out sampling from snow tracks could also further knowledge of ecological connectivity. So how are different populations connected to one another? To understand, maintain, and restore conservation, snow leopard and Eurasian lynx conservation throughout the world. They really mean any organism can be restored. So if you think about it in this sense, within population genetics, really within any biological study, some of the most expensive and time-consuming portions is collecting that data. Okay, it is very, very costly, very, very expensive to have all the field techs, to have the amount of people hours, to actually get this DNA in a form that is usable. So by using these non-invasive sampling methods, which have been used in a variety of ways, there are baited traps, so they will have some way of attracting the animal to some trap, but instead of trapping the animal, right, instead it is collecting DNA. So think of doing this with a bear and finding some method that really lets them rub up on these traps, right? So they could rub their body up, that gets hair, that gets skin cells, and from there they can collect that eDNA, all without ever interacting with the animal directly, without changing its behavior, without harming it in any way. And what is incredible is from this paper, they were actually able to identify individuals from this eDNA. They were able to say there are 12 individual polar bears that they have collected eDNA from using their tracks. Okay? So think about that. We can understand the individuals. They were fully genotyped, meaning that they extracted the full genetic information and they were able to utilize it. So we can track individuals. We can track connectivity, right? Say we find an individual, it's individual AB72, because we don't name animals, we name them just with, Elon Musk kid names. And then you find AB72 in a different population. Right, it moved over. You extracted the tracks from a different population and you found AB72 genetic material there. Great. Well, now you know that, all without needing to capture AB72 in that other population. You just found it via the hair, via the skin tracks, via a marking, right? So what I'm saying is that this methodology will enable us to do so much more for wildlife conservation, and it is also being used for anti-poaching methods. Think about this. You could swipe a suitcase coming into a country of origin. From that swipe, you could run a very quick diagnostic that says, hey, we are detecting polar bear on this suitcase. We are detecting reptiles. We are detecting Indian star tortoises on this suitcase. Perhaps this person has been in contact with endangered species of high conservation concern. So really, this paper, this video, is more of a love letter to eDNA more than anything else. And if you want to find any more information, the links are in the description. I hope this was a good overview without too much tangential excitement. But yeah, if you want to like, subscribe, share, comment, do all the things that YouTubers tell you to do, and subscribe to me on Patreon for as much as a cup of coffee a month. It goes a long way to supporting science communication here on YouTube, Instagram, and really everywhere that I do science communication. So with that, I'll leave you. Have a wonderful day.